A very good afternoon to one and all present here. I, Dr. Doel Bhattacharya, Assistant Professor of the Department of Chemistry, takes great pleasure to introduce the keynote speaker for today's valedictory function. She is none other than Dr. Aruna Kode ma'am. She is the Technical Officer of the Department of Nuclear Applications in the International Atomic Energy Agency, Vienna, Austria. Welcome, ma'am. Ma'am, are you there?
Yes, good afternoon to everybody. I'm Aruna Korde here. Yes. Are you able to hear me? Hello?
Yes, ma'am, we are able to hear you. Thank you, ma'am. I will now proceed with the formal proceedings of the session. Let me introduce our chief guest. Let me introduce our keynote speaker for today's session to all. I take this pleasure to introduce our ma'am, Aruna Kode ma'am. Hello, ma'am. Am I audible?
Yes, now it's okay.
Okay. So, Dr. Aruna Kode ma'am has joined IAEA as a radiopharmaceutical scientist at NAPC Department of Nuclear Applications in 2018. She has M.Pharmacy and PhD degree from the University of Mumbai, India. And she has also completed her postdoctoral research fellowships in PET centers from the Yale School of Medicine, CT, USA, and Wake Forest University, NC, USA. Before joining to International Atomic Energy Agency, she worked at Baba Atomic Research Center, BAR, premier research institute in the nuclear sciences in India, as a scientific officer, where she was heading the radiopharmaceutical evaluation program, along with quality assurance responsibilities of radiopharmaceuticals produced and supplied to hospitals all over the country. Over 25 years of experience in clinical research, development, and deployment for clinical use, including the planning and development of related facilities. She worked as a key member for Pharmacopoeia and radiopharmaceutical committees in India. In her current role at IAEA, she is responsible I'm sorry for the technical interruption. In her current role at IAEA, she is responsible for providing technical assistance to member states for capacity building, human resource development in RI production and radiopharmaceutical areas. Her other activities in this area are coordination of IAEA research projects, publication of GMP guidance documents, and the other relevant professionals. She also coordinates the activities of IAEA networking group of women in the radiopharmaceutical sciences. Ma'am, we are highly privileged to have you with us in the platform on ICNHTCS, that is a international conference with us. And now I would hand over the technical proceedings to our convener, ma'am, Dr. Ratna Sarkar, ma'am, who will welcome the speaker for today's session.
Good afternoon, ma'am. I, Dr. Ratna Sarkar, Head of the Department of Chemistry and convener of today's International Conference on New Horizons and Trends in Chemical Sciences 2022, welcomes all the dignitaries and the participants across the globe on the virtual platform. It gives me immense pleasure to begin this auspicious occasion by heartily welcoming our Honorable Chief Guest, Dr. Aruna Korde, ma'am, Technical Officer, International Atomic Energy, Vienna, Austria. We are highly honored to have you with us. With no further delay, madam, we request you ma'am, to start the session on the topic radiopharmaceuticals, current status and trends. All the participants are impatiently waiting to hear you. Thank you, ma'am.
Good afternoon, everybody, and many thanks for kind introduction. I think I will keep my video off if you don't mind, or if you allow me, because I think there may be some technical issues of not having the clear audio. As far as possible, I will try to keep my video on, but otherwise, just excuse me if I put this off in between.
Ma'am, it's perfectly
first,
fine.
Okay.
You may please
Thank
proceed
you.
as your convenience.
Okay, thank you so much. My heartiest congratulations to Indian Analytical Society to put together the very good program. And I'm glad to be here interacting with you all. And thank you for the opportunity. So as it's for the Analytical Chemist Association, analytical chemistry has always been the part of the radiopharmaceutical chemistry. And that's why I thought it's very appropriate that if I speak about radiopharmaceutical, which is the field in which I have been there for all my professional life. So I think my topic is acceptable to all, and I will be giving the brief status, what is a current status of radiopharmaceuticals and what is the trends in this field. So please allow me to share my presentation. I hope you are able to see my presentation. Hello? Are you able to see my presentation?
Yes, madam.
Oh, okay. Is it in full screen mode? Because hello?
मैम फुल स्क्रीन मोड करना पड़ेगा आपको, फुल स्क्रीन मोड
Okay.
करने की जी।
Okay. Yes, now I think it should be okay. Okay, so as I said, you all are analytical chemist or analytical scientist, and you are used to the spectrum of electromagnetic radiation, and you use it for all material characterization or molecular characterization. So this electromagnetic spectrum ray has a very high range, starting from the radio waves, which are kilometer long, then the microwaves, then IR, visible, UV. And beyond this comes is X-rays and gamma rays. And these gamma rays have the shortest wavelength, and as you all know, as the wavelength becomes shorter, the energy they carry increases, and more penetrating this electromagnetic radiation becomes. So everybody knows about the X-rays in medical imaging. So gamma rays, which are also similar to X-rays, but which are only emitted in nuclear reactions, and so they are more energetic, and they are used in medical imaging. So what is a basic difference between the X-ray imaging and the gamma ray and the nuclear medicine imaging, which uses the gamma rays? In X-rays,
this meeting is being recorded.
In X-rays, the radiation source is outside the body. That is the field called as radiology. And when the radiation is attenuated and it is detected on the X-ray film, you get the image depending on the attenuation of the X-ray. And when it is a 3D image, we call this a CT or computer tomography. Whereas in diagnostic nuclear medicine, what happens is the drug formulation containing the radionuclide, that is a radiopharmaceutical, which is injected into patient. It gets localized due to the physiological process, and its path and localization is detected using the suitable detectors which are outside the body. So hence it's possible to get the functional information, that is the organ functioning, or even the information of biochemical processes that are happening inside the body through this nuclear medicine imaging. And so hence the imaging equipment and the radiopharmaceutical, both are the two main important components of any nuclear medicine procedures. So as so this is which are the radionuclides of choice for the radiopharmaceutical. So we all know that gamma rays has a longer or larger penetrating power. They have the longer range. They can pass through the all medias, and they can be only stopped on the a very high side material like lead. So it's easy when the radionuclide is inside the body, and if it is emitting the gamma radiation, it's easy to detect from the outside detector. So for diagnostic imaging, the radionuclides preferred are the gamma emitting radionuclides. Whereas if you want to do a radionuclide therapy, means where the radionuclide in the radiopharmaceutical formulation is taken to the particular organ or the particular tissues, and mainly it is used for cancer, because if the particulate emitters, which are beta minus, or the alpha emitters, or the Auger electron emitters, they have a very short range, like millimeter range, and they deposit their energy in that range, and they cause the cytotoxic effect, that is they kill the cells within that vicinity. So these radionuclides, that is beta minus alpha particles or the Auger electron emitters, are preferred for the radionuclide therapy. So what modalities we have? We have two basic diagnostic imaging modalities. One is called single photon computer tomography, where the radionuclide, of course, this is a gamma emitting radionuclide because it is for the diagnostic. So this gamma emitting radionuclide that emits a single gamma ray during their decay, and and this is detected by the suitable equipment called as SPECT camera. And this is the most used example is the radionuclide has to be suitable to this energy, which is so the radionuclide should emit the energy which is suitable to be detected by the existing SPECT camera equipment. And the most used diagnostic imaging radionuclide. Hello, is there any problem?
Yes, ma'am.
Hello.
I think there is some kind of interruption, ma'am, in the signal. I'm very sorry to interrupt you at the mid of the session. Ma'am, kindly please see that your slides are not shifting, I think.
Okay. Up to which slide you could see?
Ma'am, it is only the introduction slide, the title slide.
Oh my God. Let me stop sharing this and then I will again.
Yes, ma'am, please rejoin.
Yeah.
And start sharing again.
Okay. Is this visible now?
Yes, ma'am.
Hello.
It's visible,
Hello.
ma'am.
Okay, let me try to go to the next slide.
Ma'am, put it on the slideshow mode.
Yes, but it is just a minute. Is it okay now?
Yeah, yes, ma'am.
Yeah, are you able to see the electromagnetic spectrum slide?
Yes, ma'am. Yes, ma'am.
Okay. Okay. So sorry about it. As I said, I think I just talked about the electromagnetic spectrum and the X-rays and gamma rays. Gamma rays are emitted by the nucleus and they have more penetrating power. So gamma rays are used, as we all know, X-rays are used, and similar way the gamma rays are used. The modality that is diagnostic nuclear medicine, which uses the gamma rays, and the difference I talked about is the X-rays imaging and the gamma ray imaging. In X-ray, you have the source of the X-ray outside the body. In gamma ray, you have the radionuclide injected in the form of a radiopharmaceutical formulation, and then that's why it gets accumulated based on the physiology of the body, and its path and its localization is detected using the suitable detectors which are placed outside the body. So what are the radionuclides that are used, that are the choice for the radiopharmaceutical? As we all know, gamma rays has more penetrating power. That's why they are able to come out of the body when it is injected in the radiopharmaceutical formulation, and we are able to detect them from the outside detector. So this, for diagnostic imaging, the preferred radiation are the gamma emitters, whereas when it comes to radionuclide therapy, we would like to, what is required is the deposition of the energy of radiation within the vicinity of the target of interest, like a cancer cells. So the particulate emitters like beta minus or alpha emitters, which have a very millimeter range, that is, they travel only small distance and they deposit their whole energy there. So like for example, even the Auger electron. So they are the radionuclide which emit these radiations are preferred for radionuclide therapy. So we have two kind of diagnostic nuclear medicine modality. One is called single photon computer tomography, where the radionuclide used is a gamma ray, a single that emits a single gamma, and it is detected by the equipment called as SPECT camera. So currently the available SPECT camera has a range between 100 to 300 keV. They are able to detect that range very effectively. And the most used radionuclide is the technetium-99m. And the other modality, which is which gives a more resolution, means better images, and it is also possible to do the quantification, and that is called as a positron emission tomography or the PET imaging, where the radionuclide used are that they are the positron emitters. So what happens is the positron positron emitted from the radionuclide, which is localized inside the body for radiopharmaceutical, after the injection of a radiopharmaceutical, that positron travels a small distance depending on the energy of the positron, and it annihilates with the adjacent electron and gives the two 511 keV gamma rays, and they are exactly at 180 degrees because of several reasons of nuclear phenomena, and I won't be able to go detail into it, but you can see that they are exactly 180 degree apart, and they are detected by the coincidence detector. You can see the camera, which is like a round detector ring, and this PET imaging has emerged as a new modality called as molecular imaging, because you are able to see what is happening at the molecular level or the biochemical processes through this PET imaging. So our IAEA database of nuclear medicine centers, these are the database which is from the middle or the low and middle income countries. This doesn't include the high income countries. And they everywhere, these nuclear medicine centers have become a part of the medical tertiary care or the and then in India also, our Atomic Energy Regulatory Board India site shows that there are around 360 nuclear medicine centers as of 2021. There's information. So what is the radiopharmaceutical? So radiopharmaceutical, as I said, the functional imaging, functional part comes from the radiopharmaceutical. Functional imaging part comes from the radiopharmaceutical, as well as the targeted therapy of certain cancers. The responsible component is the radiopharmaceutical, because it's like it's a drug formulation, and a radionuclide and the carrier molecules are the two main components of radiopharmaceutical. The radioisotope component determines the application, whether the product is used for diagnostic or for therapeutic or for both the purpose. As I said, the gamma emitter, or whether it is the radionuclide uses beta emitter, depending on that, the application of radiopharmaceutical will be there. But and the physical decay characteristics, such as what is its energy, what is the type of emission, half-life, as well as what is the chemical properties of that elemental properties of the radionuclide, they all are important while selecting the radioisotope. The carrier molecule is the biological determines the biological properties and currently various carrier molecules. So as there has been the development in biomedical sciences, the more and more new targets have been identified, more and more small molecules, different type of peptides, proteins have been now identified as a possible markers, or these all have been tried to be labeled with the suitable radionuclides, and that is how it's not easy to have the radiopharmaceutical design, because what you need is the optimum accumulation and the desired retention of radionuclide only in the target organ. If you want to take if you want to image heart, your formulation should go only to the heart and not to the other organs, like it should be cleared from all other organs. Specifically, if you want to target the cancer cells, it's very difficult, and then it should be, so the clearance from the all other target tissues.
Excuse me, ma'am.
Yes. Yes.
Ma'am, I think there is again some interruption in the network, that your slides are not changing.
Oh, okay. So you saw up to which slides? Are you able to hear me clearly?
Ma'am, we are able to hear you clearly, but the now we are on slide 8.
Okay.
Ma'am, you may continue from here.
Okay. So as I said, the radionuclide is the and the carrier molecule are two main important components. Radionuclides are various ways of production of radionuclides. They use either the nuclear reactors or accelerators or cyclotron, where in accelerators. Shall I think, are you able to see only the? Okay. So the accelerator and cyclotron, they use the charge particles, energetic charge particle, to bombard onto the target. And but there are several other factors that need the consideration when radioisotope is used for radiopharmaceutical product development, which is radiopharmaceutical product development is a long process, involves a lot of resources, and it's necessary that the one should realize the feasibility of the production in sufficient quantity and in desirable quality, as well as the process needs to be compliant with good manufacturing practices, as this is considered as an active pharmaceutical ingredient of the radiopharmaceutical product, which is used in clinic. So these are the few examples of radio isotopes for SPECT imaging, but I won't be able to go in detail into it. But then, as I said, technetium-99m is the most used and most used radionuclide because of several advantages. One is it is easily available in the form of a generator. Then its chemistry is very versatile, and it has a convenient half-life for imaging, suitable energy for SPECT. So what is the generator system? Just in brief, that this is a kind of milking a cow. You have a generator which you place in the hospital radiopharmacy, and this generator contains a parent nuclide, that is molybdenum-99, which has a 66 hours half-life, and its daughter radionuclide is technetium-99m, which has a 6 hours half-life. So there is a kind of equilibrium which reaches around 23 hours. So every 23 hours, when the molybdenum is decaying, you will see the maximum buildup of technetium. And there is in 1950 1958, one lady Margaret Greene, she developed the process. She developed the procedure for separation of this molybdenum from the separation of technetium-99 from the molybdenum-99, and this lead to the development of this generator, where now you can see that the current generator, you use the high specific activity molybdenum-99. It is immobilized in the form of a sodium molybdenate onto acidic alumina column, and the pharmaceutical grade, you just put a saline and you get the pharmaceutical grade technetium-99 for your radiopharmaceutical preparation. So what happens in nuclear medicine, in hospital radiopharmacy, is you have the cold kits, that is like a drug formulation, and you have a generator, and you have this kind of hot cell where for having the protection, radiation protection, as well as the pharmaceutical compliance, and then you use the technetium eluted from the generator. You take the cold kit depending on what you need. If suppose you want to image brain, you take the cold kit which is for the brain imaging, and you mix, and as per the manufacturer's instruction, you do the preparation and you do the quality control, and your product is really ready for patient use. So why this technetium? Because technetium has versatile chemistry, and hence it's a kit-based radiopharmaceutical preparation is possible. If it forms a different complexes, and the characteristic of that complexes means it's not, it has been designed, the formulation is designed such a way that, suppose if this is called as technetium MeB, you all know that a positively charged complex can get into heart because of like sodium potassium pump of the heart. So this is a positively charged lipophilic complex, so it goes into heart. This is a neutral lipophilic complex which goes into the brain, that is technetium HMPO. You have the phosphonate ligands complex with technetium-99. So phosphonate being the affinity for the bone, the MDP, that is methyl diphosphonate, it is a very good bone imaging agent. Similar way, you have all different radiopharmaceutical for renal imaging, for liver imaging, for gallbladder, for RBCs, and for heart imaging. And the technetium is the most used. 80% of the diagnostic nuclear medicine is being carried out using the technetium compounds. So when it comes to the positron emission tomography, as I said, this is the most means all clinician, they would like to have their images using the PET camera. So this is since it's a high resolution and quantification is possible. So what we have different type of radionuclides for positron emission tomography. We have the short half-life biological elements like carbon, nitrogen, oxygen, and fluorine-18 radioisotopes. They are of, if you see, their half-life is very, very short, and that's why they need to be produced at site or at least in the vicinity of the hospital. So, but what are the new emerging radionuclides? They are the radiometers, and they have a lot other advantages. I will talk about it in the next few slides. So what is the most used PET radionuclide is F18 fluorodeoxyglucose. It's a glucose analog, and it is called as molecule of millennium because of its clinical significance, because of its clinical demand. The whole technology has brought this, made the cyclotrons compact, and the whole, since this is positron emitter radionuclides emit high energy radiation, so they demand you cannot just use them in the simple hot cell, and you cannot use them by hand. So you then demand the remote synthesis procedure. So whole technology got developed in last say last 3 decades, and now you have the having a cyclotron at the hospital is become very common. And so this FDG is the most used, being a short half-life, it be 110 minutes. It needs to be produced locally, or at least in the vicinity, because if I'm in India also, almost all major cities, metros have at least one cyclotron in the city, and they cater to all other hospitals which have the PET CT cameras. So first, what is done is the F18 radionuclide is produced in sufficient quantities in cyclotron. Then the radiosynthesis is carried out. The product is tested for quality control, the physiochemical, radiochemical, pharmaceutical quality control, and then it is injected into patient. So FDG, being a glucose analog, it gets into the cell similar to how the glucose enters into the cell. However, its further metabolism is interrupted due to structural difference between the FDG and the glucose. So it gets metabolically trapped inside the cell. So wherever there is more glucose uptake, such as in growing tumor cells or in the brain, which definitely need more glucose, you can see more FDG uptake as well. And then since it is not further metabolized, it gets trapped, and F18 is detected using the PET camera, and you can clearly see this red color, that is, and this is like a slice of the image. So it has FDG has several applications in cancer, in oncology, in neurology, and in cardiology. So also gallium-68 is another important PET radioisotope, and more than 10,000 scans are performed only in Europe. In India also gallium-68 is available. So what this gallium-68 has an advantage is it can lead to the specific different type of radiopharmaceutical design is possible, and this is also produced using a generator at the hospital, and you have the different synthesis module like this to prepare the radiopharmaceutical. I will talk, and this is a very important radionuclide for theranostic imaging. I will talk about it in the next few slides. So what this gallium-68 is produced in the generator form, but this generator has, as the demand of gallium-68 is growing, there is the cost of the generator is going, the cost is increasing, then there is a less availability of this generator. So what is alternative is, if you have a medium energy cyclotron, you can produce this gallium in large quantities, and you can do the more number of patients compared to what you can do by eluting the generator at the hospital. So, in India, the Brit cyclotron that is about DAE unit at Calcutta has recently produced this gallium from medical cyclotron, and it is available locally, at least in the Calcutta region. So when it comes to therapeutic radiopharmaceutical, which is a very important field and which is a growing field. So as I said, you need a particulate emitter radionuclide. Most common one is a beta emitters. So the iodine is the synonymous with the nuclear therapeutic therapeutic radiopharmaceutical since ages, because the man knew how the iodine gets incorporated into the thyroid. It's a physiological process. So similar to any non-radioactive iodine, if you have a radioiodine isotope with a beta emitter, it also gets accumulated into the thyroid, and most of the thyroid cancers are curable by using the iodine-131 therapy. So these are the some of the phosphonates ligands, or the where the bones, you can say the bone seeking ligands labeled with therapeutic radionuclide like lutetium, samarium, lutetium 177, samarium 153, or rhenium 188, or alpha emitter, that is radium 223 chloride. So these are all bone seeking agents. Why bone seeking agent? Because they carry this radio beta emitters or the alpha emitter radionuclides to the bone, and they are used for the bone pain palliation, that is to relieve the pain in the terminally ill cancer patient where they have a lot of bone metastasis developed at the later stage of the cancer. And there are specific therapeutic radiopharmaceutical for treating cancers, and these are the very much emerging, and they the most used currently is lutetium 177 because of its physical properties, its convenient half-life, so that logistically to produce, you can produce at one place and you can ship it at one other place. But this needs a suitable infrastructure and very much trained human resources. So it's preferred to have a ready-to-use dose of such radiopharmaceutical. And there are commercial companies supplying these products all over the world, and in India, DAE unit also supply such lutetium labeled radiopharmaceutical products, which are very specific for certain type of tumors, that is mainly the neuroendocrine tumors and also for the prostate cancers. So what is in trend or the emerging thing in therapeutic radiopharmaceuticals is use of alpha emitters, and that is called as targeted alpha therapy, TAT. And it's lot of efforts are currently being put for development for these mainly focusing on some few radionuclides. Actinium-225, which has 10 days half-life, is one of them. And this actinium, currently the actinium demand over all over the world is much more than what can be produced. So there are different production routes have been explored by many of the. Yes. Again, there is a problem.
Yes, ma'am, there is a problem with the slide sharing. Ma'am, can you
Okay.
please come on the current slide?
Which just a minute. Okay, are you able to see the picture slide with a image?
Yes, ma'am.
Okay.
It's slide number 23, emerging radionuclides.
Okay. So this is a actinium 225 labeled with this. So this, as I said, there are different routes people are trying to explore to meet the demand of actinium 225 for the clinical studies currently, as there are very promising results shown by some of the compounds labeled with actinium. That is this is a prostate cancer agent labeled with an agent for prostate cancer treatment labeled with actinium, and you can see the patient with lot of metastasis, and after certain treatment it is cleared, and that's why there is a very much interest. So what is this theranostic radiopharmaceutical? So as I said, there are diagnostic radiopharmaceutical, there are therapeutic radiopharmaceutical. So what is theranostic? Theranostic is like you are able to use the same radiopharmaceutical design. That is, if you see, and this is mainly for the cancer cells, because what happens is the cancer cell expresses certain particular type of proteins or the markers on its surface when the cell becomes cancerous. And so these, if you identify a specific molecules which are able to specifically bind to this overexpressed proteins on the cancerous cells, like this. So this is a, let me take the pointer. So this is a particular molecule, more particular protein. Cancer cell is overexpressing compared to normal cell, and you have this kind of antibody, or this is a target molecule which you want to target, and you have this particular specific molecule which binds to this target which is overexpressed onto cancer cell. You use particular design, means to combine the radionuclide. So your radionuclide is here, you put some spacers or the chelator to hold this radionuclide. So this is like you have a bifunctional agent here which holds a radionuclide at one side, and your specific carrier molecule on the other side, and you are able to take this radionuclide specifically to cancer cell. So if you are able to use the same design, means same carrier molecule, same design, and only change the radionuclide, then what you will be able to do is you will be able to diagnose the particular type of a cancer using the diagnostic radiopharmaceutical. And once you get the image, once you get the expression of the cancers, even, and you will be able to treat those. So it is like, it's very useful for screening, characterization, treatment selection, response monitoring. So it's like a personalized targeted therapy, and this is very successfully applied for neuroendocrine tumor and prostate cancers. So coming to the end of my talk, what are the radiopharmaceutical? That they are the preparation containing radionuclides, and they differ from the conventional pharmaceutical products. They have the limited shelf lives due to the associated radionuclide. So hence majority of the product use is subjected to local production. Like any other pharmaceutical product, you cannot keep them produced for a longer in larger batches and supply it world over. It is not possible. So they are subjected to both radiation protection and health or pharmaceutical regulations. They need the parametric release, because some of the products have such a short half-life that you need to use them before you complete all the quality control tests. So you need a good quality management system to assure that what products you have produced are of good quality and suitable for clinical use. And there is a limited availability of the current education in the radiopharmaceutical field. So it's a multidisciplinary activity. You need the physicists or nuclear engineers for production of radioisotopes. Then you need the chemist, radiochemist for radioligand development, cell biologists, pharmacologists to evaluate the product at the biological level. Then you need the preclinical, you need to carry out the preclinical studies where veterinary medicine people or the medical physicists, they contribute. Then once you have the good radiopharmaceutical formulation, then you need to develop the process which is a GMP compliant, and then you need to do a lot of regulatory clearance. You need to collect the data, put it for regulatory approval, and then only it goes into the patient. So it's a long way, but it's very it's similar to any of the pharmaceutical process development, but with more challenges and complications. So nuclear medicine, so this is the extract from nuclear medicine in Europe. So you have currently a lot of scope to do the imaging and therapy with the different kind of radiopharmaceutical. And the if you see the growth of the radiopharmaceutical field from 2013 and what is going to be expected in 2025, the major growth is in the therapeutic radiopharmaceutical, the blue part here. And so that's all from my side. If you have any question, I will be very happy to answer. And I'm very sorry about
Thank you,
the
ma'am.
technical difficulties. And thank you.
Thank you, ma'am.
Yes.
Thank you, ma'am. Ma'am,
I request you all to stand on your respective seats. Thank you.